Thank you, everybody. First of all, thank you for the SACS team for organizing this wonderful meeting. You know, the best comes last. So we are looking forward to a great session. There are some housekeeping rules. So please, for the companies, keep the 3-minute rule for talking so we have some time for questions. And for the audience, please take your seats. Be attentive and support the companies by posing good questions and showing your interest. I'm Tomáš Rajkofer from 4051 Ventures. This is a Basel-based small venture fund company, trained as an MD, PhD, interested in many, many different therapeutic modalities. So with that, let me introduce you the first company. This is Meddenovo from Lyon, and the speaker is Ilke, Ugur, Marion. Please go ahead.
Good afternoon, everyone. My name is Ilke. I'm the co-founder and CEO of Meddenovo Drug Design. And at Meddenovo, we develop cyclic peptide therapeutics using our own AI tools. AI is just a tool. What really interests us, what is our ice cream, is cyclic peptides. And we find them extremely advantageous compared to small molecules. They are more selective, hence less toxic. And compared to biologics, we can produce them in higher scale up because finally they are chemically synthesizable. Venture Capital 101, everything with great potential comes with great challenge. And one of the main challenges of cyclic peptides is that they are very expensive. So this traditional trial and error in drug discovery should be optimized by computational approaches. This is perfect, but up to now there are no computer approaches or AI algorithms specific for cyclic peptides. And this is exactly what we did actually. We developed Mexa. It's our proprietary AI engine. Mexa does something quite interesting. If one of our co-development partners approached to us with a target without no data, no information, no literature, no science, based on only the target structure, we can find a starting structure so that the project can initiate. Mexa is not an idea, it's not a project. We have successfully applied it several next-in-class drug discovery projects. One of the very notable ones is a radioligand we did, which reaches nanomolar level of inhibition. And with the contract we signed with Daiichi Sankyo in November, we are also advancing our own pipeline. As I mentioned, we are advancing our own pipeline, and Mexa is not commercially available to external partners. It can only be reached in internal solutions. And who is making these applications? This is our team. I co-founded Meddenovo with Antal. We have been working together since the last 13 years, not only for cyclic peptides or AI, we have been developing different solutions to some challenging problems in drug discovery. And now we are not alone. We started alone, but we are now a team of 8 employees. We are also supported in our advisory board. For example, Troy Dare, ex-VP from Vertes, is giving us strategic advice. Our board members are also supporting us in financial decisions. Finally, we raised 1.7 million pre-seed previously, and with that amount of money, we were able to build an engine which designs de novo molecules. And we also build our own computer cluster, which brought me to the end of my time that we can run many projects in parallel independently. And now we are raising 7 million, and I am happy to discuss the details how we will be advancing our pipeline with this seed round. Thank you very much.
Thank you for the presentation. One of the challenges of the peptides are a very short PK. Have you find through AI a way to increase the PK of peptide by maybe having some selected 3D structures in your cyclic peptide? Thank you.
All the properties of peptides, they can be adjusted with how you build the structure and the combination of everything you use between natural and non-canonical amino acids. And these are all project-dependent selections. Whatever has to be optimized depending the final therapeutic application. We've selected a number of biases as Meddenovo team, and based on these biases, we run our simulations.
All right, thank you very much. Our next speaker is Vincent Forster from Nerai Biosciences, and he's talking about novel CRISPR editors with high in vivo activity.
All right. Good afternoon, everyone. Vincent Forster, CEO of Nerai Bioscience. Nerai really leads the precision in genome editing, and I'm excited today to show you how we can unlock the full potential of this technology. Genome editing really redefined modern medicine, the way we used to do it and the way we're doing it now. So instead of focusing on symptom and really treating the consequences of disease, genome editing gave us the key to really be able to treat disease at its root and provide cure even before the symptoms arise. But these tools are not today fully efficient. And one of the reasons is that CRISPR-Cas needs to recognize a short DNA sequence on the DNA, which must be close to the mutation. And this sequence is called PAM. And this PAM requirement really limits the use of CRISPR-Cas to a minority of genetic disease, typically 20%. So of course, people have seen that and have created novel variants, which we call pan-relax variants, but these come at the cost of a reduced activity and increase off-target, so decreased safety. We're taking a more challenging but more rewarding approach by designing specific CRISPR editors that are bespoke to each targeted mutation and targeted DNA sequence. And with that, we can unlock new indication with high efficacy on target and low or no off-target effect. And we build those by creating the most sophisticated CRISPR engineering platform to date, combining directed evolution at scale, fully automatized with high throughput characterization and an AI model. This accumulates decades of academic research, and today we can create the editors that we want with the characteristic that we choose, which are typically high performance, high specificity, and of course high safety with low off-target. Our first lead candidate is called NB301 and is directed towards a rare urea cycle disorder disease called citrullinemia type 1. This disease really matches all the criteria for an accelerated and streamlined clinical development all the way to approval, being a severe treatment with no drug available and clear regulatory incentives. Once this editor would be approved in this indication, we are also aiming at targeting 7 other rare diseases by just swapping the guide RNA and accessing these diseases in a cost-efficient fashion. And this is today possible thanks to all the beautiful new regulatory incentive that the FDA is putting forward for rare disease acceleration. We are today completing our proof of concept in vivo, and we expect to reach patients by 2027. This modularity of the platform has also attracted some traction from the large pharmaceutical company, and we intend to also partner with those in non-core area. And backed by competitive non-dilutive funding and a pre-seed of 1 million, we're now raising a 6 million seed round to really complete the full preclinical studies and move the product closer to patients. With that, I thank you very much and happy to answer your questions.
So, thank you very much, Vincent. Questions from the audience. For me, this type of targeting is always a question, can you really reach in real life those cells which you want to edit? So with the citrullinemia, can you give us an example? What is the outcome in vivo at the end?
Yeah, we have already done in vivo experiments now. We also can prove that we can edit 30% of the hepatocytes, which are the targeted cells, right? And 30% is actually a very, very strong editing rate in vivo after systemic administration. The in vivo targeting is done to the vector, right? Which we use lipid nanoparticle for targeting the liver. And we don't innovate or create any new IP into the lipid nanoparticle. We work with the best in class, which have already been saved and tested in patients. And we can really stand on the shoulders of giants which have done and performed this technology before us. Thanks.
Thank you very much, then, Vincent. Our next speaker is Antal Szalay. Please take the podium. And this is about oral medications, which is interfering with a metabolite, which has multiple consequences in humans, including potentially neural influences in the neurodegeneration.
Good afternoon. My name is Antal. I'm the CEO and founder of Ultimate Medicine. Together with my team, we detected a novel brain-toxic metabolite that directly disrupts key brain functions and is likely involved in cognitive impairment and dementia. So we work on delaying the onset of dementia significantly. Delta-Valerobetain is a likely driver of cognitive decline. What you see on the left-hand graph is schematically depicted data that we generated analyzing blood samples of the flagship cohort from Australia, ABLE, led by Colin Masters. What you see on the y-axis is cognitive performance and functional performance, and the red line depicts how steeply patients decline with elevated levels compared to low levels, the blue line. So this metabolite directly affects mitochondrial and neuronal function, increases with age, it is in all of us, and is elevated in patients with Alzheimer's disease. The metabolite is produced in bacteria. So this is what you see on the right-hand side of the graph, of the slide, is a graph that depicts the elevated levels of a critical enzyme that is elevated in Alzheimer patients and contributes to delta-Valerobetain or DVB production. In our foundational Nature paper, we exposed mice to increasing concentrations of DVB. That's what you see on the left-hand graph. These are the brain concentrations. And you see dose-dependent cognitive decrease in 2 standard mouse models. This is the T-Mason novel object recognition test. So this supports also the associative data we had from the patient cohort. We decreased to a certain extent the metabolite levels in individuals using antibiotics, but I guess we all agree this is not a targeted and specific long-term strategy. That's why we chose, and this is what you see on the left-hand side, to alter the function of bacteria to decrease production and thus the systemic circulation, stopping the increase and normalizing elevated levels. So we did a structured drug discovery approach. We have discovered first hit clusters and moved now into our bacterial model system. That's what you see on the right-hand side, inhibited DVB production. Let's imagine how could a targeted profile look like. So this is more meant as a vision based on the current understanding and the data compared to A-beta therapies. You see we have an Alzheimer-related targets, first-in-class oral administration is possible. The efficacy estimated based on the data is very high, more than 400% delay compared to 30 to 35%. A high safety profile, it's just orally administered and simple to diagnose, thus expected to be easily accessible and taken in, so adopted. At this point, I want to thank the team with its deep drug development expertise from early discovery through clinical and commercialization, contributing significantly to health outcomes with more than 15 approved drugs. Our work is supported by top KOLs. So we closed our first pre-seed round, and it was led by the renowned Alzheimer's Drug Discovery Foundation. Now we are raising a seed round to develop an industry standard candidate and then plan to have an early proof of mechanism in phase 1 based on our biomarker panel. I thank you so much for your attention, and I'm happy to answer any questions after the session.
So, thank you, Antal. Maybe for completeness, if you allow me, I start with a question because 5 years ago, a number of papers came out, and I have read actually your paper at that time. This was for the audience. This was one of the first demonstrations of the famous gut-brain axis with a real metabolite. So it's a real fantastic academic work. Now, at the same time, there was also publications about that the same compound is also obesogenic, so it's causing body weight, but also that it influences neonatal and early brain development. So are you not too late with your therapy?
Thank you for your kind words. It's true. So internally, we speak about this metabolite as a lifespan switch metabolite, and there is evidence and also on our hand unpublished data that is involved in our early stages of neurodevelopment. From our understanding, when we become older, so when we age, our metabolism changes and the impact of the metabolite on the system changes, it increases, and so it is context dependent. So that's also why we say we don't want to eliminate it, we want to normalize the levels. And it's true, we clearly are focused here with our first indication on Alzheimer's because we have very strong data on this. Nevertheless, clearly there is the possibility to scale this into other indications and treat other chronic disease potentially. So this is what you see, this is really emerging biology, and we worked in a very structured way the last years on it.
Thank you very much. Our next speaker is Otten from TheraPPI Biosciences from Geneva. And he's talking about a new way of addressing a dysregulated MAP kinase target. So Luke.
Thank you, everyone. I'm Otten, CEO of TheraPPI and co-founder. And I'll give you two numbers. 150 clinical trials and 50 programs for the same target, RAS, evaluating RAS inhibitors. That's a huge number. All these drugs, all these programs end up as inhibiting the signal, and it goes very rapidly to resistance. We want to change that. We want to improve the fight against drug resistance by dysregulating the signal. So let's dig in. On the left, you see that that's the RAS-MAP kinase pathway, which is activated in 40% of all cancers. And no wonder, huge number of pharma and huge number of programs have been done, and especially on the RAS. They all do at the very end, they inhibit the enzymatic activity of ERK, and rapidly you have resistance, probably because that's a propensity of the pathway to get reactivated by biological nature of it. We approach the problem differently. We use at the very end the gatekeeper ERK. We keep it active, but we dysregulate the signal through inhibition of protein-protein interaction between ERK and an unexpected protein called MIDT8. And with that, that brings us a number of advantage, among them the lack of induction of resistance, but also direct tumor cells killing, and also an induction of anti-tumor immune response. Where we want to go with our company today, we are at an early stage discovery company. We want in 2 years to be able to get our preclinical candidate and move a bit more than a year after to be ready for the first in-human trial. For that, we need 5 + 5 million Swiss francs as a seed round. And we see, given the appetite of the pharma, the possibility to have the first window of opportunity at the beginning of IND enabling studies. I want you to have 3 things to remember. First, we want to change the fight against drug resistance by dysregulating the RAS-MAP kinase signaling. We have ambition to beat all the drugs that inhibit this signaling, including the RAS inhibitors. And we plan to do the best possible drug development so that it shall attract interest of the pharma to collaborate with us or to acquire us. So I thank you for your attention. I thank you all our supporters, and I'm very happy to answer your question.
Thank you, Luke, very much. Any questions from the audience? So I must admit I was interested in MID88, and there are certain mutations, not infrequently. Would your approach work against the mutated MID88?
Yes, it's an excellent question. And indeed, there are different mutations with MID88. There are mutations that are inherited immunodeficiency. That is in one part of the domain. And there is let's say, the intermediate domain. And there is another domain that binds ERK. And that's this part, which is a different one. And so that's in answering your question, we don't inhibit the innate immunity ability of MIDT8. We keep it normal. And we have, by the way, mutated this domain on the MIDT8. And so to validate with this part of the target, and we have mutated the ERK part as well. So because when you do PPI, you need to do the genetic validation on both sides. It's more complicated.
So thank you very much, Luc. We need to move on.
Yep.
And our next speaker is Guido Gualdoni, who is talking about a program called Cadence. So we are very curious about hearing what that means.
Hi everyone, I'm Guido Gualdoni. I'm CEO and co-founder of Epitome Therapeutics, a company that we just launched out of EMBL with a next generation epigenetic editing technology. And you're actually one of the first ones to hear about it. So when you think about it, most diseases are not caused by broken genes, but by genes operating on a wrong level, by actually dysregulation. And existing modalities are very binary in how they interact with the target. So we think that there's a huge potential in orchestrating gene expression across multiple targets. And we think that gene regulation is the missing modality in medicine. It has a huge impact on diseases such as haploinsufficiencies, metabolic disorder, and eventually also cancer and aging. So Epitome's proprietary Cadence technology that we just launched out of EMBL is the first technology to durably enable activation of gene expression in an unprecedented precision. It's considerably safer than other approaches. And Cadence will turn gene regulation into actual medicine. So how does it work? My co-founder, Jamie Hackett at EMBL, he developed this tool based on a catalytically inactive Cas9, as you can see here on the left-hand side, that recruits epigenetic effector molecules to a locus of interest. He developed 13 different epigenetic effectors that induce very specific histone marks. As you can see here in the middle, compared to the blue bar, a standard CRISPR-A that very strongly activates gene expression, certain combinations of the epigenetic effector molecules are even stronger. But importantly, we can also fine-tune the expression to exactly the level that we're interested in. So we can durably activate gene expression, tune it, and can really control gene expression. Just briefly walking you through one of the diseases that we're interested in, haploinsufficiency, Alagille syndrome. Haploinsufficient because one allele is mutated and one is wild type, and there's 50% gene dosage. If you now were able to upregulate the gene dosage to 2 to 3-fold, we could cure this disease that currently has no treatment option. And actually, there are not many therapeutic applications that could move towards this. Just briefly walking you through the team, myself, I'm MD PhD by training. It's my second venture, and I've already conducted multiple VC rounds and also big pharma licensing deals. Jamie Hackett, the co-founder, is a established key opinion leader in this area, and we've onboarded very early on drug development expertise for the delivery aspect, as we're also delivering in LNPs. Briefly walking you through the next milestones. We have, since our foundation 6 months ago, already raised 900K through CLA and grants. We've established a lab now and are fully operational. And we're raising a 2 million pre-seed round, of which 1 million are already committed to reach drug candidate nomination, as we're also able to obtain another 4 million in non-dilutive funding in Austria. So with a total 6 million, we are aiming to reach preclinical candidate nomination by the end of 2027. So with this, thank you for the attention, and I'm happy to take questions.
Thank you, Guido. I'm sure there are some questions from the audience. I think with gene regulations, especially if you are putting epigenetic marks on that, my question is always, will this marking survive cell division? Is it inheritable to the next one? And of course, targeting is an issue. Can you really hit all the necessary cells what you want?
That's a brilliant question, and actually that's one of the very specific USPs of this technology, as it's the first epigenetic editing technology that can actually persist over mitosis. So there's certain combination of marks that actually persist over mitosis, and it's the first technology that can therefore durably activate genes. That's something that's entirely novel in the space. And the delivery aspect, again, very similar to Nerai. The first programs that we're launching are directed to the liver, as we don't want to invent the wheel. Liver targeting LNPs are very much established, and we're not stacking delivery risk on technology risk here.
Thank you, Guido. Excellent. There's a question out there. Luca, please.
Quick question. X chromosome silencing, could you potentially have a bias in the therapeutic applications?
Say again? Can you?
The X chromosome silencing, so could you potentially have a bias in the therapeutic approaches?
So that's something that we would need to look at if there's something that's entirely silenced, whether we can overcome this. So this is something that we would have to empirically test. Thank you for the question.
Thank you. With the next company, we are still staying in my favorite town, Vienna. So RIANA Therapeutics and Anna Orlova is talking about how to inhibit STAT5.
Good afternoon, everyone. My name is Anna Orlova from RIANA Therapeutics, and it's my great pleasure to present to you today our first-in-class STAT5 oligomerization inhibitors. So the problem we're working on is the resistance in FLT3-positive AML. So a lot of patients in AML really relapsed very fast, and once they do so, there's not much that can be done to prevent this disease progression. And the problem is that we don't have good inhibitors on the market. The problem is that those inhibitors cannot eliminate leukemic stem cells that are driven by STAT5. So current treatments are mostly for FLT3-positive AML, is mostly around FLT3 inhibition or venetoclax BCL2 inhibition. And both inhibitors have really great clinical and commercial value, except that the patients eventually relapse. And the problem of the relapse that the STAT5, the node exactly in between FLT3 and BCL2, remains active and drives disease progression. And STAT5 is a very known validated target in leukemia, but also other cancers. And the problem is targeting STAT5, it has a dual role. It can function as a dimer, which presents the STAT5 essential functions in blood cells, liver functions, and so on. But STAT5 is also a very potent oncogene, which is usually mediated, and it was discovered in our lab by oligomer formation, when there's 4, 6, 8, and more parts of like the STAT5 forming clusters. So whereas our competitors tried and validated STAT5 as a valid therapies by developing STAT5 PROTACs, what they did not succeed is to create a therapeutic window, because they also eliminated essential functions of STAT5, which are essential. And what we're trying to do differently is to target the protein-protein interaction, which mediates oligomer formation. And by this, we will eliminate cancer cells without harming healthy cells, because dimers will remain intact. So our first indication is FLT3 AML, which is chosen not by randomly. There is a perfect indication because STAT5 has a directly activates STAT5, it drives the resistance biology, and there's clearly defined patient population. So far, we finished our hit identification, we validated our hits, and are currently developing them further to nominate a lead candidate soon. We validated that our inhibitors actually work better in venetoclax-resistant cells, proving this opportunity to target this. And our first tool compounds, we already validated they're active in reducing tumor formation in mice, even in the unoptimized stage without unoptimized PK. So in our terms of our timeline, we follow in classical drug discovery platform. So far with our pre-seed round, we managed to get the middle of hit to lead stage, and we're currently raising our seed round approximately 2.5 million euros, which is already partially committed. And in terms of our investment, we're currently raising 1 million equity, which we aim to leverage with a public grant funding. And that should bring us to our lead candidate nomination with in vivo proof of concept. So please talk to me if you're interested in this opportunity. And thank you for your attention.
Thank you, Anna. Questions from the audience? So we understand that STATs themselves are tough to target.
Yes.
But there were a couple of approaches to that. So most notably, Janssen had dopamine agonist or dopamine antagonist, which is called Pimozid, which for a while people touted as a STAT5 modulator.
Yes. It is unfortunately a kinase inhibitor.
Okay.
Yes, so it works indeed on STAT5, but it's one level upstream, so it targets a kind of STAT5 mediated kinases.
All right. So
We use
then
it as
your approach is really unique.
Yes, we really want to target it directly.
Thank you very much. So our next speaker is Mamta Chabria from Tandem Therapeutics, and she is addressing a very specific field, mechanobiology, targeting matrix under pathologic conditions.
Thank you, Thomas. Good afternoon. It's my pleasure to be here. My name is Mamta Chabria, co-founder and CEO of Tandem Therapeutics, spin-off of ETH and PSI, so very local. We have one singular mission at Tandem Therapeutics, which is to target the extracellular matrix to halt fibrosis progression, which is the current gap. We do this by developing mechano-targeted peptide drug conjugates. But before I get into that, fibrosis remains a major unmet need, which is growing and doubling incidence rates, specifically in the lung. What we know now is that it is aging and injury-driven, and it has a huge organ burden that it can affect different organ states and cause fatalities. The key part in fibrosis is the extracellular matrix is exclusively deposited uncontrollably, and this results in organ failure. Now, one key insight into this is that the extracellular matrix actually displays mechanical signatures that are only present in the fibrotic matrix, but not in the healthy tissue. And this is something that Tandem Therapeutics is leveraging with our understanding of the ECM to target by developing peptide drug conjugates and be a first-in-class biology of mechanomedicine approach in this area. So I want to actually take a moment to tell you about this molecular strain sensor, which is the central technology or the core technology on which we are developing our peptide drug conjugates. What you see here, we are able to use a peptide-based sequence to essentially help us read out the mechanical strain state of a fibrillar protein called fibronectin that is highly expressed and upregulated in fibrosis, in organ fibrosis, and also oncology or fibrotic tumors. What we are trying to do with this molecular strain sensor is it allows us to read this strain state, and we are able to map this strain state to fibrosis across different scales of assays, acellular, cellular, but also as a next step, we are able to actually take this and home it into the fibrotic tissue, as you can see here, where we have conjugated the peptide with a radioligand moiety and delivered it and homed it into the fibrotic lung where it shows up, whereas in the healthy lung, we do not see any uptake. What is very important also is that we notice a very strong positive correlation of the presence of peptide in the lung with the fibrotic score, so the degree of fibrosis that you can see in the cohort in this particular sample on the plot on your right. So with that, with the core technology of the molecular strain sensor, we are developing peptide drug conjugates by linking validated clinical payloads to bring this bioconjugate into the fibrotic core, which allows us to actually fulfill the gap of having sustained activity and safety at the fibrotic matrix part. We are developing this now with into a new class of antifibrotic, mechano-targeted antifibrotic pipeline, and we are backing our series seed round based on the recent milestones we've achieved, not only across targeting with our peptides, but actually showing a PK/PD connect with our PDCs in progressive lung fibrosis model, and actually most importantly, the human translational model, the precision cut lung slices, where we see a superiority of our targeting approach to the untargeted approach. So we've been part of an accelerator program and had over 2.5 million non-dilutive and with the accelerator program. And currently we are raising a 7 million seed round to help us essentially take our lead PDC towards DC in lung and liver and expand on the platform, especially being the TPP for radioligand use. So of course, this is not a singular effort. We, the co-founding team has several years of experience having worked together, especially in the field of mechanobiology, and we are supported by a great operational and advisory team. So if the story of outside the cell approach in fibrosis excites you, please reach out. I would love to tell more.
Thank you, Mamta. Maybe we have time to one question, please.
Great presentation, thank you very much. How will you administer this product? Will it be a systemic administration?
No, we are aiming for a subcut for fibrosis.
Okay, but it will be systemic?
Yes, of course, yes.
And a very naive question, I don't know which indication you'll go into first, but if it's lung, a lot of these patients will have fibrosis in other tissues, which will be great because I think you'll target all of the fibrotic tissues, if I understand correctly. It may also be a challenge to measure safety and efficacy during those sorts of studies. Have you already thought about that and discussing with clinical experts?
Yes, absolutely. So you're absolutely right. There can be several comorbidities, not necessarily only limited to fibrosis. We have seen definitely target engagement, which is very fibrosis focused when we are targeting in the lung. So we do see binding in other organ systems, but we do not see a target engagement as of now. But this is an ongoing, let's say, efforts in trying to be very selective on the mechanobiology, which is sequestered only to fibrosis.
Thank you, Mamta. We need to move on. So our next speaker is Alain Vertes. He's coming from OncoNex-Remunity, a multinational, multi-target cancer biotech.
Okay, brilliant. And I very much appreciate the introduction and the invitation to speak today. So you're right, exactly. This is exactly what we're developing, a multinational company, a multinational oncology specialist biotech, which we want sustainable. And how do we want to do this out of de novo, basically? So basically, we assemble first the critical success factors, the critical elements of this engine. The team needs to be competent, needs to be international, it needs to be willing to work well together in trust. The second aspect of this is obviously to have access to the world leading research centers and ecosystems and KOLs, and obviously a brilliant portfolio of products that provides us with a balanced options. So this is our strategy. Let's go back directly to the footprint. Footprint we have established first in the US near MD Anderson Cancer Center. We are formally supported by Portal Innovation and TMCI. In France, we incubated by Servier and partners, their incubator. We are formally supported by Paris Saclay Cancer Cluster. We are very lucky to have this support, and we are also building a presence now in Hong Kong, thanks to our friends at Invest Hong Kong. This way, we have access to the US, we have access to Europe, and we have access to Asia. What do we want to do? We want to, of course, access KOLs who have demonstrated they can do it. John Emack particularly brought to the market 2 drugs last year and is willing to work with us. What we have assembled is a portfolio, as I mentioned, balanced, best-in-class program, because we want to manage a risk we can manage, that is operational risk and commercial risk. And we have selected clinical programs that are fully de-risked and clinical validated. So we have 2 phase 1 programs, big pharma legacy, a tri-specific T-cell engager, HER2, but with a twist, much safer than the other inhibitor similar to this. We have a small molecule FGFR inhibitor with a great profile, very, very clean, providing us a very significant advantage to over what was developed and approved. And obviously, the best in class is fully de-risked because completely clinically validated, but we have first in class approaches as well out of our own technology programs. So this way, by advancing all of this, we really create this engine to bring new products to patients who otherwise would have no hope. We bring therapeutics that are targeted. We bring therapeutics that are small molecule, TCs, but what we are really doing is creating a de novo, a sustainable biotech company. We have a variety of indications. The positioning is very clear. Second line to NR2, fantastic market. NR2 is about 15 billion by 2030. We can be first in class in China with our FGFR inhibitor. We have a very clear commercial pathway that was fully validated by our KOL. Again, we are world renowned. We are looking for fundings, our Series A, to do this very quickly on this. I have 12 seconds left, but basically we are looking only for 25 million and the company that is licensing is keeping only 5% of the equity. Thank you very much for your attention.
Thank you, Alain. Questions from the audience? I think what you presented and at a very high level is very impressive. Can you really finance this with 25 million? And how do you prioritize if you are limited with the funds?
Well, that's obviously the killer question, and I was not expecting anything less, actually. But you're right, this is very little amount of money. Sorry, I can't go back. But basically, our approach is really to focus first on China. China first with the FGFR inhibitor. It's a small molecule, it's very cheap to produce. We have a first-in-class profile there. We can go to after cholangiocarcinoma. We can go after bladder cancer. Great indications, fully accepted by the FDA with an FGFR inhibitor, which does not have the same profile as we have. We have very little retinopathies, but the single percentage when the other FGFR inhibitor provides like 12% of retinopathies. We have a very, very good compliance with this drug and 35 patient dose for each. So we have really good demonstration of this.
Right.
So China is our answer.
First China and cholangiocarcinoma.
Yes.
Thank you.
Thank you so much.
So our next speaker is, here he is, Kaido Kurrikoff from Tartu, Estonia. And he will talk about non-LNP mRNA delivery into tissues other than liver, which I think is very exciting from multiple perspectives.
Hello, my name is Kaido from Vectiopep, and I will be speaking about cold tumors and immuno-oncology. So immuno-oncology market is dominated by immune checkpoint inhibitors. Immuno-oncology therapeutics combined represent a 50 billion annual revenue. However, this 50 billion is used to treat just 20% of all cancer patients, which have the hot tumor phenotype. So in Vectiopep, we are looking towards the cold tumor and the huge market this represents. Vectiopep is a drug delivery technology, and its uniqueness is defined in 3 points. Vectiopep is precision delivery of messenger RNA very selectively into the immune cells. In the left here, I'm presenting more than 90% selective transport of mRNA into the immune organ spleen. And this precision transport is achieved by using unique chemical entities. Vectiopep is using peptides instead of lipids. In the second pane, due to this precision transport of messenger RNA into the immune cells, we are able to actually achieve 10 times the amount of the killer T cell activation. And these preclinical data have been validated in 3 different species, plus human PBMCs. And finally, third, on the right side, by targeting just one cancer antigen, we are able to overcome cold tumor in a mouse model. So this is colorectal cancer model. So in humans, our primary program targets multisatellite stable colorectal cancer, which is a prevalent form of the cold tumor colorectal cancer. And due to Vectiopep precision delivery into the immune cells, we are completely avoiding the off-target toxicities from the liver accumulation. And secondly, in the middle pane, unlike LNPs, we are able to drive unprecedented amount of the T cell immune activation. And unlike CAR T, we are not just targeting one single epitope from one single cancer antigen, but we are mounting a multi-attack against multiple tumor antigens simultaneously. And finally, third, on the right side, we are not just targeting cancer antigens, but mRNA design also allows targeting the tumor stroma antigens, which is the driving force of the cold tumor phenotype. Vectiopep just raised 1.2 million pre-seed funding, so we are fully funded to finalize the development compound, and we will achieve the lead asset lock and TRL5 in the second half of 2026, and after that we will launch the IND enabling studies. And here today, already now, we are seeking for investors who would help us through this preclinical development of our CRC cancer indication. Thank you very much for your attention.
Thank you, Kaido. Questions from the audience? Luca, please.
Great presentation, thank you. If you deliver antigens to the spleen, you could potentially induce tolerance. Do you have evidence that your system can be used to induce tolerance to antigens?
We are inducing immune activation. That is what we have measured. And because we are avoiding actually the off-target accumulation to liver, that is actually the reason why we are so confident in stating that we are actually doing the opposite of inducing immune tolerance.
So, Kaido, very briefly, would you be able to share the nature of the mRNA, what you are delivering?
The RNA is composed of actually a series of tumor antigen and stromal antigen epitopes, basically very simple stating, put on top of, next to each other. So one single messenger RNA contains and encodes for many, many different tumor antigens from the same molecule.
All right, thank you very much again. We need to move on. So, Bjørn-Yoshimoto Walden from Akasi Pharma, Copenhagen, Denmark, is talking about somatostatin receptor, small peptide agonists. About pain.
Yeah. Thank you. My name is Walden. I'm the CEO and co-founder of Akasi Pharma. And at Akasi Pharma, we are making pain therapeutics for some of the very difficult types of pain. Initially, diabetic peripheral neuropathic pain. This is a chronic complication of diabetes that affects about 100 million patients worldwide. And one of the main problems is that it's really, really difficult to treat. Within the first 3 months of treatment, more than 50% of the patients, they discontinue. And within the first year, it's more than 3/4. The main problems and what patients are actually saying they need in new therapies is something more efficacious, so something that actually works. They also want something longer lasting and with a faster onset of action. Then they emphasize convenience with fewer pills and safety and tolerability. To tackle this, we're looking towards a target called the somatostatin receptor 4. And there are several reasons for this. One is that it's peripheral in its action, meaning we don't need to get our assets into the brain, so no CNS side effects. It's also tolerance resistant, meaning it's actually a good target for chronic treatment. And it's been validated clinically in a phase 2A trial, so it actually alleviates pain in the patients we're trying to treat. The way we go about modulating this is with a small peptide modality, and the main reason is to circumvent some of the issues seen by small molecule competitors. It's still small enough that it's cheap and fairly easy to produce, and we've been able to make versions that are compatible with weekly or even less frequent dosing. We've shown effect in various animal models. Seen here is the spared nerve injury model of neuropathic pain. You can see our drug compared to gabapentin, a clinically relevant comparator. Now, with this big unmet need and very big market, there are, of course, many things in development. I want to focus on just a couple of these. One is the broad class of ion channel blockers. Here, Vertes is leading the field. However, clinical evidence suggests that our target can deliver significantly better efficacy than they can. We're also longer lasting with fewer pills. The other one I want to highlight is other somatostatin receptor 4 agonists. And here, Eli Lilly's story is the most interesting. Again, we're longer lasting with fewer pills, but where we really shine here is on the safety parameters. While Lilly actually validated the target with this program, they also saw drug-drug interactions with metformin. And if you're trying to treat diabetics, that's not ideal. They also had a lot of side effects, including gastrointestinal and CNS. They've since discontinued their program, while we, on the other hand, have actually already shown some tangible and real advantages on these parameters. We're now in lead optimization with an IND slated for 2029. This will also facilitate our move into the clinic, where we'll finish a phase 2A by 2032. Right now, we're looking to raise 15 million euros to get through phase 1. To do this, we have a small but very dedicated core team, and we've surrounded ourselves with expert advisors and consultants to help fill some of these gaps we have. Thank you, and I'm happy to take a question.
Thank you, Walden. Questions from the audience. So what we were impressed, of course, was this recent move from Vertex and some other companies, Navigate Therapeutics and so on, NAV 1.8. So when you need to compare them now side by side because of the fantastic valuation of that and the enthusiasm about that, what would you say is your advantage, also given the pharmacokinetic differences?
Sure. So one thing is, of course, the long acting, which a lot of patients are looking for. Another thing is that Vertex's phase 2 trials with diabetic peripheral neuropathy, I mean, it wasn't really better than pregabalin. So, I mean, there was an effect, but nothing better than what's on the market today. And that's what patients say isn't good enough. If you look towards Lilly's phase 2A trial, numerically, they had about a 25% better efficacy. And that's some of the best we've seen in clinical trials.
All right. So wishing you all the best. Thank you very much.
Thank you very much.
Our next company is Therapeutics. And Mrs. Dominique Doyen is talking about difficult to target GTPases.
Okay, so we are a startup based in Munich, and we want to specialize in developing GTPase inhibitors, which are difficult to drug targets and usually that are dysfunctional in disease. And of course, our lead program is looking at KRAS mutated cancer. So this is our team with the two co-founders with experience in R&D, business development and operation. And well, we are, of course, looking at KRAS because it's 25% of 25% of all cancers are driven by KRAS. And of course, it's also looking at very lethal cancers like pancreas, non-small cell lung cancer, colorectal. And it's a market way above 10 billion. So what is the problem with KRAS? Where it's a validated target, 2 drugs have been approved, but looking specifically at 12% of the mutations, which is a KRAS G12C, sorry, but it's not working. So basically, here this is a KRAS G12C, and it is only 12% of the patients, which means that 88% of the patients do not have any treatment. So what is doing the field? So as we hear also before, there are a lot of people looking at KRAS specifically inhibitors, looking at all the different mutations, or looking at multi-inhibitor with Revolution Medicines leading the field here and where we expect phase 3 data for pancreas during this year. But what's happened is that usually you get something like 40 to 60% of the patients that are already non-respondent, and then afterwards you get patients that are resistant. So there is something to do and to improve here in terms of development. And for that, you look back to the, let's say, tumor progression. And of course, you go to this KRAS pathway, a little bit simplified. And if you look at this pathway, you have 3 main branches. And what's happened with these oncogenic signal is that usually the RAF-MEK signal will do the induction signal for the tumor growth, while the other side of the pathway, which is driven by PI3K and RAL, is going to be responsible for the metabolic reprogramming. And RAL is one of these GTPases which has completely been overlooked, and this is what we are looking at Tuari. And we think that it is really a superior approach because it's not only driven by KRAS, it's also driven by the tumor microenvironment. So basically what's happening is that if you block KRAS, well, you will block partially KRAS pathway, but if you block RAL, you have really a way to be much more stronger in the inhibition, but also you will deal with the resistance that these patients will see. So the differentiation is, of course, to look to these patients that are non-responding and becoming resistant to KRAS therapy. And, well, we do have validated hits. We want to move to the proof of concept in vivo. For that, we are raising 2.7 million with already 1.1 million, which is committed by our existing investor. Thank you very much, and happy to get any questions.
Thank you very much. So given the broad competition in this field, I understand your target is different.
Yeah.
Is the chemistry different, or is the expected outcome then different?
Well, it's very interesting because, of course, you do have some, let's say it's a GTPase, it has some similarity with KRAS, but actually we are looking at different mode of inhibition. So it's, of course, small molecule, but we have completely different side of addressing this inhibition.
What would be your first indication ultimately where you would really obtain proof of concept?
Well, most of us, we will certainly look at the, let's say, three main indications: pancreas, lung, and colorectal, but specifically looking at these patients that never responded to the traditional KRAS treatment and the ones that are becoming resistant. That will be our first criterion, because we have really here, we see in this type of mutations really effect of our compounds that we don't see with the other, like Revolution medicine. So
sounds fantastic, ambitious.
Hopefully.
Thank you very much for the
You're
presentation.
welcome. Thank you.
So our next speaker is Jason Golan from Elaris, and we are back to Vienna.
Perfect. So thank you very, very much for the invitation and for being here for the talk. My name is Jason Golan, and then I am indeed from the adopted city of Vienna. After a career of about 20 years working at Valneva, also a Viennese-based global vaccine company, we created a spin-out, my co-founder and I, back in 2025, focused on Clostridium difficile. And Clostridium difficile, or more importantly, in fact, hospital-acquired diseases, as we know, is one of the most persistent and also costly problems today when it comes to the modern healthcare system. C. diff is a potentially life-threatening diarrheal disease, impacting about 1 million cases per year. So largely impacting immunocompromised elderly and individuals in hospital settings. Despite the global unmet medical need and extreme cost to healthcare systems, today there is no preventive treatment for C. diff. Despite this, there's approximately a $3 to $6 billion market opportunity. So the need is there, the market opportunity exists. However, no solution is there just yet. As mentioned, about 1 million patients annually, so largely elderly, in which there is approximately a 10% mortality rate. So individuals above the age of 65 who contract C. diff end up succumbing within approximately 4 weeks. The current care or first-line treatment is antibiotics. And despite the fact that it's associated very strongly with strong recurrence rates, so about 30 to 50%, a number of the large pharma companies involved in vaccine development over the years have tried to develop a toxin-based approach. So going after the neutralizing toxins, in fact, in the gut, with limited success, up until Pfizer, in fact, who demonstrated in the subpopulation that the vaccine did have a clinical benefit. Elaris is the only company in later stage development here with a dual mode of action. So recognizing that toxin neutralization is indeed the foundation, we licensed a phase 2 asset from Valneva, where my co-founder and I were coming from, and combined it with a second colonization and blocking component. This is an idea on paper. In fact, as mentioned, we have strong clinical data supporting strong results, as well as a preclinical proof of concept. Moving forward, we're looking to raise today 20 million, allowing us to enter into phase 1 within the next 18 months and really unlock the future, the next big inflection point for this program. Recent transactions show elderly vaccines, adult vaccines are extremely attractive, with 300 to 1 billion euro transactions, money transactions over the past 12 months from big pharma companies. With that, I want to thank everyone for your attention. As mentioned, we're looking to raise about 3 million euros in the short term, enabling us to reach our IND, followed by clinical entry. Thanks so much.
Thank you very much, Jason. Questions? 2, 3.
Thanks for the presentation. I think it's important that people realize it's a spore-forming bacteria, and that's one of the reasons that it's so difficult to get rid of. So I really like your approach where you're doing two things. Your vaccine approach, which is one shot, but recolonization is really what you need to be targeting as well, and that's exactly what you're doing. So yeah, one shot, killing two birds, that sounds great. So I like the technology. Thank you.
Thanks.
Thank you very much. So the target population is elderly, right? So is the idea going to be, okay, everybody above 55 gets that as a vaccine, or how do you think of the?
Yeah, that's a great question. And that's actually probably one of the most common questions we get. Where would the vaccine be used? And of course, universal vaccine would be the holy grail. So universal vaccine for individuals above the age of maybe 60 or 65. Most likely, maybe one day it goes in that direction, similar to other elderly vaccines like pneumococcal and such. But certainly focusing on elderly individuals with immunocompromise, living in elderly homes, people going in for elective surgeries is a key population. Oncology patients on chemotherapy whose systems are suppressed. Actually, we see a very high incidence rates for C. diff impacting their treatments. There's a couple of very isolated specific cohorts, which would be a clear recommendation in the first instance. The idea would be to expand this out to a broader recommendation, community-based type recommendations.
A great presentation. Fecal transplantation, Ferring as a product on the market that was developed by Biotics, wouldn't be that an alternative as well?
Also a great question. So there's a number of solutions. So as mentioned, antibiotics, in fact, is the solution, but also the problem, because that's what's driving the high recurrence rates. Once you get into this vicious cycle of infection treatment, reinfection treatment, the only real option, in fact, is these more or solutions like fecal transplantations and the like to really reset the microbiome. So here, our proposal here is not necessarily is really actually to inhibit the cases of C. diff before they occur. So if you're that far down the line where you need a fecal microbial transplantation, you're very, very far down the line. You've gone through probably months of repeated cycles of the disease, and here, from an overall impact on patient, cost to healthcare system, this in fact is probably a patient which has costed the healthcare system, the payers, hundreds of thousands of euros. So that's in fact exactly what we're trying to avoid.
Thank you very much, Jason. We need to move on. Our next speaker is, thank you. Our next speaker is, where are you? Simone? Simone Sabbione from Meliodays, and she's talking about an IUD device for menstrual pain relief.
Hi, my name is Simone Sabbione. I'm the CEO and co-founder of Meliodays Medical. And at Meliodays, we're developing the first local non-hormonal pain management solution for menstrual pain. This is Tina. And since Tina first got her period at the age of 13, she's been suffering such severe menstrual pain that she had to skip out from school, later on from university, and work recurrently. Now she's 30 years old and she's trying to get pregnant. Again, the chronic inflammation of her uterus is preventing that. And this is precisely what we are targeting to solve with Melio-1. Melio-1 is a small implantable device that can be put directly into the uterus, just like a conventional IUD. But instead of releasing hormones, we are releasing a very low dose of anti-inflammatory pain medication, which is targeting the prostaglandin upbuild that actually causes this recurrent and chronic inflammation of the uterus. And this is the pain results from underlying conditions such as endometriosis, adenomyosis, or uterine fibroids. So those are all patients we can help with our device. And of course, maybe also importantly, our platform is currently being explored to be also used for pre-ART treatment, so also for IVF optimization. We asked 800 women if they would be interested to use our product, and 45% responded with a clear yes and 25% with a maybe. Now, the market is super large because so many people are affected, but we are only planning to capture 1.5% of the TAM. Since we have the competitive products in the market are mainly hormonal and they come with severe side effects, we have a unique opportunity here to have the first product that's non-hormonal applied in the uterus. Also, we are targeting a 260 euro price in front of customer, which enables us to have a direct or payer entry to the market. We achieved significant milestones by filing our RIP in 20 markets by now, closing our pre-seed round with 1.2 million, and we're currently conducting preclinical studies showing 20 times fold higher concentrations in the uterine tissue compared to systemic circulation. And we had our pre-IND meeting with the FDA already in December, aiming now for EMA scientific meeting in June. And this helped us to have a clear pathway throughout the preclinical studies to know exactly what's needed for an IND meeting and to move forward for clinical approval. We are aiming to launch our product in 2030 in the US, which is our first market. The team has a combined experience with medical, scientific, commercial, and scaling expertise, and we're currently raising 2.5 million in order to advance our product and get to IND readiness. So if you're interested, please approach me, and also happy to answer questions.
Thank you very much, Simone. I'm sure there would be questions from the audience.
Thank you. So we had quite a lot of discussion. Very interesting indeed. Can you just comment for the broader audience about other indications? Because I think women's health is until now not very well targeted and treated. So
yes, so that's actually one of the challenges we are having, because women's health has been under research and under studies for a very long time. So this is very new to actually, like, this chronic inflammation of the uterus does a lot. As I said, it first results in pain. It can be endometriosis, adenomyosis. There's so many different underlying conditions there. But also this chronic inflammation is critical for conceiving. So in the whole fertility process, for instance, in IVF treatments currently, once there needs to be a diagnosed repetitive implantation failure before medical professionals look at the uterus actually. So before that, it's always assumed it's a sperm, it's the egg, or it's a combination. But then when you look at the uterus, it's diagnosed to be inflamed, and then usually the treatment is antibiotics, assuming it's a bacterial inflammation, but there's no real proof for it. So we actually assume that by targeting this inflammation, there could be much more potential in healing the uterus sustainably over a longer period of time, and actually learning what this would actually do. And this has never been tried before.
Very interesting. Thank you very much.
Thank you.
Our next speaker is Kevin Hollywood from Hemastax GmbH. And this is about bleeding disorders, targeting von Willebrand factor destruction.
Yeah. Good. Thank you all for sticking around. So I have the pleasure indeed to present you Hemastatics. So I'm the CEO and one of the co-founders. So Hemastatics is a relatively young company. So we incorporated earlier last year. We are a spin-off of the University of Leuven and are also supported by Base Launch, amongst others. So our mission is really to transform the field of bleeding disorders, starting with our first lead program. So our first lead program, HMX-1, is an antibody targeted at ADAMTS13. So ADAMTS13 plays a pivotal role in the primary hemostasis. It's the delicate balance between VWF, so the von Willebrand factor, and ADAMTS13 as its modulator. So what we've been doing in the past year, in addition to building a team, is expanding the preclinical package, also the IP. We've refined the clinical roadmap and also been progressing towards a DC nomination. We're currently here to start the fundraising for the next stages of the company, which includes starting the IND-enabling work, and of course to bring this molecule to the clinic, ideally by the end of 2028. A bit of background on the disease pathology. So our disease, or our patient population, suffer from pathological unfolding of VWF. So in a normal individual, the high molecular weight VWF is a multimeric clotting protein, which regulates the bleeding in a very tight and elegant fashion. However, in our patient population, there is this pathological unfolding of the most active format of VWF. And as a consequence thereof, ADAMTS13, which can be considered as a molecular scissor, is basically excessively cleaving VWF. As a consequence thereof, you'll have trouble if a bleeding episode happens, because you won't be able to stop the bleeding. And the process by itself, this excessive cleavage, has also an impact on the angiogenesis, and as a consequence thereof, you increase a risk of bleeding. This is obvious in patients that have a genetic predisposition. So you have patients that have a specific mutation that lead to this unfolding of VWF. And then you also have patients that have an acquired syndrome. So this typically happens due to shear stress. So this shear stress basically is induced either by, for example, a heart pump. So you have that flow in the blood that unfolds VWF. But it can also happen with other cardiovascular indications or, for example, also with aortic stenosis. So it's a fairly, let's say, elaborate patient population. What we've been looking at is an antibody that specifically binds and inhibits ADAMTS13 activity. And by doing so, you can really restore the normal activity of VWF. So this has been a decade of work at the University of Leuven. We believe we have a very robust basis for continued value generation. So the previous work has generated a lot of IP already. So we have 2 patent families that are either approved or close to approval. We continue to building on that during the current lead optimization. Preclinical package, I showed the data to some of you, ex vivo data and in vivo data in a calf model. We're also working on some mouse model data as we speak. Clinical roadmap has been defined to find a way to leverage the genetic component as a first indication and then move this towards the other indications tied to acquired VWS. And lastly, we've also been doing some market analysis that show that this molecule from a pipeline and a product perspective can certainly leverage severe or premium pricing in severe bleeding disorders and also has consequently the appropriate potential in terms of market. And our current focus is now on this DC nomination and clinical positioning. And what we are, of course, here today to further discuss with you is on one hand to show you how compelling of an asset our first lead program is, but then also, of course, start to build this coalition of the willing, let's say, to further advance the company. Our current estimates is that it will take us about 20 million to wrap up the first in human, first in patient type of trial. However, of course, with the, let's say, smaller tickets, we can also significantly advance the program in the years to come. And this is something I'm happy to further discuss with you. Thank you.
Thank you, Kevin. Maybe this time I can start with a question. Yes. So there's actually an acquired condition where there are autoantibodies against these enzymes.
Yes, of course.
And it is having 2 consequences. One is they are natural antibodies. So wouldn't it be nice to get them and use them as a therapeutic?
Yeah.
On the other hand, this is a condition which is severely affecting thrombosis.
What is important there to highlight is that we consider the risk of TTP for sure. So in our preclinical models, in our animal models, we have not seen such signs because we specifically looked for it. That can be to some extent tied to the shear stress that is in the blood. And then as a consequence, you don't have these ultra large VWF. Of course, we continue to focus on that. And I think there's 2 ways to go about it. On the one hand, you want to refine your therapeutic window vis-a-vis the duration and also the depth of inhibition of ADAMTS13 to kind of find a balance between the optimal, let's say, therapeutic solution to resolve the bleeding and avoiding the risk. On the other hand, that's something that we're also exploring. You could advance partial inhibitors that are then, let's say, keeping the safety margin and then allow for longer dosing, which would unlock the prophylactic application as well.
So in such a situation, what is the optimal half-life for an antibody? You may not want to be too long, but also not too short.
And so in the data that we currently have, the antibodies are about, in these calves, they're about 4 or 5 days, which gives you a nice window. Of course, if you do the extrapolation, because we've been doing some modeling, then you typically end up with a normal half-life of an antibody of 3 weeks, which might be a bit excessive. So from our perspective, we're probably looking at something that has more of a half-life of a few days. So then you're either in the, let's say, a type of an engineered antibody or some format that can do the trick. But again, if you have a partial inhibitor, the half-life might not matter as much.
Thank you.
Yeah, good, thank you.
Thank you, Kevin. And please welcome our last speaker of the session and of the day. And this is Jan Degen from KES Biosolutions.
Hi. This is for the camera. Hi, I'm Jan, and I'm here with something else entirely. I'm not here with a therapeutic or an indication. We are building a drug discovery platform, which is run by enzymes. How does it work? We combine our enzyme with a lot of starters, extenders, and the like, and we access a vast chemical space that hasn't been accessed before. How do we translate this? How do we find a ligand? How do we find a candidate? So, of course, we start with some in silico prediction model. We do overnight one-pot reactions for the molecules, and we scan these immediately and repeat this until we meet the threshold for the molecule that we want to have. We call this combination of, enzymatic production and fragment-based discovery. We call it evolutionary fragment growing. How does this turn into business? Well, we believe that we can run the company profitable by just with just running on these fees and success fees and lead optimization. Of course, the upside is in milestone payments and royalties for the developer. We believe, because the entry fee is just 150K, that we can democratize drug discovery a bit by firstly giving AI way more molecules to pick from, and then widen the customer base down to foundations, networks, groups, and even single individuals. And we can scale the whole process because it's an aqueous reaction just by adding more automation. We're currently looking for a 2 million raise. And in order to build, we need this in order to build our precursor library and to leverage a 1 million grant. And we want to validate the platform on 3 targets. In total, this will give us a 3-year runway, and I'm way quicker than I expected. But so far, I can tell you I've listed up some facts, so we can spend time on these. We've so far raised 1.7 million in equity plus 1.4 million in grants. We are 6 people now. We're based in Potsdam. We founded in 2022. We have 2 patents pending and still 80% of the shares in our hands. Thank you.
Thank you very much. Any questions from the audience? Yeah, maybe it would be helpful if you could just explain one particularly successful example, because this is really a different
it's completely
area.
different. I realized after I've seen the other presentation. So what we did is, we are not a spin-off of a university. We decided in 2022 that we want to build the company from scratch. So we had to redevelop everything that has been published before. So my co-founder, he published in Nature Chemistry a way of derivatizing erythromycin and rapamycin, like polyketide stuff. And so we developed everything from scratch again. And yeah, so the first milestone was to have to patent everything. And then the second milestone we had, so we have like a library of 150 completely novel molecules by now of these precursors that we combine then to generate the larger molecules.
Are these compounds actually fulfilling the famous 5 of Pfizer?
Well, they are. Some are breaking the rules, but most of them are within. So of these 400 billion, 90% fulfill them.
Okay, so they are drug-like.
They are drug-like, yes. They are drug-like. We start with smaller fragments, but of course we combine them, and we can even combine these with classical reactions just as building blocks to combine them into larger molecules.
Thank you very much.
Okay. Thank you.
And so thank you again. With this, we are closing our session. Thank you for the participation. First of all, for the speakers. We know that this is tough to present your case in 3 minutes. So thank you for being so adamant on that. And also thanks again for the SACS team for organizing this wonderful session and a wonderful meeting as well.